Revolutionizing medicine. CRISPR. Hope on the horizon. The dawn of the genetic age. The biological sciences have long operated under the paradigm of observation and minimal intervention, understanding the book of life without possessing the tools to rewrite it. The emergence of clustered, regularly interspaced short palindromic repeats, or CRISPR, and its associated proteins, particularly Cas9, has shattered this limitation, granting humanity the unprecedented ability to edit the genetic code with the ease of a word processor. This transition marks a fundamental inflection point in human history, moving medicine from a reactive discipline treating symptoms to a proactive one, capable of addressing the root causes of pathology at the nucleotide level. The implications for human health are staggering. We are witnessing the first generation of therapeutics that do not merely manage chronic conditions, but aim for curative finality. From the eradication of monogenic disorders like sickle cell anemia to the intricate engineering of immune cells to hunt solid tumors, CRISPR is redefining the boundaries of what is clinically possible. Yet, as the technology matures from the lab bench to life-saving breakthroughs, it brings with it a complex tapestry of challenges: technical hurdles regarding delivery and off-target effects, profound ethical dilemmas concerning human augmentation and longevity, and the stark economic realities of million-dollar cures. This review provides an exhaustive analysis of the current state of CRISPR technology, exploring its mechanistic evolution, clinical applications in hematology, oncology, and rare disease, its speculative future in human enhancement and longevity, and its critical role in global food security. The molecular architectures of editing, from blunt force to precision. To understand the clinical potential and limitations of current gene therapies, one must first appreciate the rapid evolution of the molecular tools themselves. The original CRISPR-Cas9 system, while revolutionary, acts as a blunt instrument, creating double-strand breaks, or DSBs, that force the cell to repair itself, often aggressively and unpredictably. The field has since swiftly pivoted toward precision editing modalities that offer greater control and safety. The canonical CRISPR-Cas9 system functions as a programmable endonuclease. It comprises a Cas9 enzyme, which acts as the molecular scissors, and a guide RNA, or gRNA, that directs the enzyme to a specific 20-nucleotide sequence in the genome. Upon binding, and contingent on the presence of a protospacer-adjacent motif, or PAM, Cas9 induces a DSB. The therapeutic outcome depends entirely on how the cell repairs this break. Non-homologous end joining, or NHEJ, is the cell's default, error-prone repair pathway. It frequently introduces small insertions or deletions, or indels, at the cut site. In therapeutic contexts, NHEJ is primarily used for gene disruption, breaking a gene to silence it. This is the mechanism behind Casgevy, or exa-cel, where the BCL11A erythroid enhancer is disrupted to release the brake on fetal hemoglobin production. Homology-directed repair, or HDR, if a donor DNA template is provided, the cell can use it to repair the break via recombination, allowing for precise gene insertion or correction. However, HDR is inefficient in non-dividing cells, which constitute the majority of adult somatic tissues, and competes with the faster NHEJ pathway, limiting its therapeutic utility. The reliance on DSBs presents significant genotoxic risks. A double-strand break is a catastrophic event for a cell. If misrepaired, it can lead to large chromosomal deletions, translocations, where parts of chromosomes swap places, and the activation of the p53 DNA damage response, which can trigger apoptosis or select for p53 mutated, potentially cancerous cells. Addressing the risks of DSBs, base editing represents a second-generation technology that chemically converts one DNA base into another without cutting the DNA backbone. Developed largely by David Liu's lab, these tools fuse a catalytically impaired dead Cas9 or dCas9 to a nickase or nCas9 to a deaminase enzyme. Base editors operate on the concept of chemical transition. Cytosine base editors, or CBEs, convert a cytosine-guanine base pair into a thymine-adenine base pair. Adenine base editors, or ABEs, convert an adenine-thymine base pair into a guanine-cytosine base pair. Because base editing avoids DSBs, it dramatically reduces the incidence of indels and chromosomal rearrangements. Research in cardiac gene therapy models has shown base editing to achieve efficiencies as high as 92% with minimal cytotoxicity, significantly outperforming standard Cas9 in safety profiles. However, base editing is limited to transition mutations, purine to purine or pyrimidine to pyrimidine, and cannot easily correct transversions or install insertions or deletions, restricting its scope to a subset of pathogenic mutations. Prime editing is the most versatile tool in the current arsenal, described often as a search and replace gene editor. It couples a Cas9 nickase with a reverse transcriptase enzyme and utilizes a prime editing guide RNA, or PEG RNA. The PEG RNA is a complex molecule that not only finds the target site, but also encodes the template for the desired edit. The Cas9 nickase cuts one strand of the DNA. The reverse transcriptase uses the PEG RNA template to synthesize a new DNA flap containing the desired edit directly at the nick site. The cell's repair machinery incorporates this flap into the genome, permanently installing the edit. Prime editing can correct all 12 types of point mutations, as well as perform precise insertions and deletions without requiring a donor DNA template or causing DSBs. While earlier iterations faced efficiency challenges, recent optimizations in 2024 by researchers at MIT have reduced error rates by orders of magnitude, from 1 error in 7 edits to 1 in 101 or even 1 in 543 in high-precision modes. This leap in fidelity brings prime editing closer to clinical viability for complex genetic disorders that simpler tools cannot address. A persistent shadow over all gene editing modalities is the risk of off-target effects, unintended edits at genomic sites that resemble the target sequence. Such errors could disrupt tumor suppressor genes or activate oncogenes. Detection. The industry has standardized around highly sensitive detection methods like Guide-seq, Circle-seq, and Site-seq to map these off-target events across the genome. Mitigation. The shift toward base and prime editing is fundamentally a safety play. Recent comparative studies confirm that large DNA deletions, greater than 100 base pairs, a common artifact of Cas9 editing, are approximately 20-fold less frequent in base and prime editing. From lab bench to breakthrough, hematology and the first cures. The theoretical potential of CRISPR has been realized most dramatically in the field of hemoglobinopathies, diseases of the red blood cells like sickle cell disease and transfusion-dependent beta thalassemia, or TDT. These conditions were the logical low-hanging fruit for gene editing because the target cells, hematopoietic stem cells, can be harvested, edited outside the body, or ex vivo, and reinfused, bypassing the immense challenge of systemic delivery. In late 2023 and early 2024, regulatory bodies in the UK, US, FDA, and Middle East approved Posgivi, or Exagemlogeno Auditem-cel, marking the official entry of CRISPR into commercial medicine. The biological strategy. Both SCD and TDT are caused by mutations affecting the adult hemoglobin subunit, or beta globin. However, humans possess a gene for a backup form of hemoglobin, fetal hemoglobin, or HbF, which is naturally silenced shortly after birth by the transcription factor BCL11A. COSGV employs CRISPR-Cas9 to disrupt the erythroid-specific enhancer region of the BCL11A gene. By breaking this off switch, the therapy tricks the body into reactivating fetal hemoglobin production, which can functionally replace the defective adult hemoglobin. Clinical efficacy data. The data from the CLIMB trials supporting this approval are robust and life-changing. Sickle cell disease, or CLIMB SCD-121. In a pivotal cohort of 30 evaluable patients, 97% achieved the primary endpoint of freedom from severe vaso-occlusive crises for at least 12 consecutive months. The average duration of this freedom was 22.4 months. VOCs are excruciatingly painful events caused by sickled cells blocking blood flow, often leading to organ damage and early death. Eliminating them effectively transforms the patient's quality of life. Beta thalassemia, or CLIMB Thal-111. For TDT patients, who typically require lifelong monthly blood transfusions, the results were equally striking. 91% of patients, 29 of 32, became transfusion independent, with durable responses observed for over 3 years. This liberates patients from the iron overload and logistical burden associated with chronic transfusion therapy. While the genetic edit is elegant, the patient journey is arduous. COSGV is an autologous ex vivo therapy. Patients must undergo apheresis to collect their stem cells, which are then shipped to a manufacturing facility for editing, a process taking months. Before the edited cells can be reinfused, the patient must undergo myeloablative conditioning with high-dose chemotherapy, or bolsufan, to wipe out their existing bone marrow. This conditioning regimen is highly toxic. Adverse events reported in trials include stomatitis, or mouth sores, febrile neutropenia, and dangerously low platelet counts. Critically, bolsufan conditioning carries a high risk of permanent infertility, forcing patients to make difficult choices about fertility preservation before treatment. The development of non-genotoxic conditioning agents, for example, antibody-drug conjugates targeting CD117, is a major research priority to make these cures safer and more accessible. The success of COSGV has spurred a race for next-generation therapies. Editas Medicine, or EDIT 301. Utilizing Cas12a, a nuclease that creates staggered sticky ends rather than blunt cuts, this therapy targets the gamma-globin promoter directly, or HBG1/2, rather than the BCL11A repressor. Early data suggests this may lead to even higher levels of HbF induction. Beam therapies, or Beam 101. Employing base editing to mimic a natural mutation, or hereditary persistence of fetal hemoglobin, without creating double-strand breaks. This approach aims to reduce the genotoxic risks associated with Cas9. Oncology, engineering the army within. While hematology focuses on fixing broken genes, the application of CRISPR in oncology is about enhancement, engineering the immune system to recognize and destroy cancer more effectively than nature intended. Chimeric antigen receptor T-cell therapy has been a breakthrough for blood cancers, but current versions are autologous, made from the patient's own cells. This process is slow, expensive, and often fails if the patient's T-cells are exhausted from prior chemotherapy. CRISPR is enabling the shift to allogeneic, off-the-shelf CAR-T therapies derived from healthy donors. The allogeneic engineering challenge is to use donor T-cells without causing fatal graft versus host disease or rejection. Multiple edits are required. TCR knockout. The endogenous T-cell receptor, TCR, must be deleted so the donor cells do not attack the patient's body. MHC class I knockout. The beta-2 microglobulin gene is disrupted to remove MHC class I molecules, making the donor cells invisible to the patient's immune system. Potency enhancement. Genes like Regnase 1 and TGF-br2 are knocked out to prevent the CAR T cells from becoming exhausted or suppressed by the tumor microenvironment. Recent data from CRISPR Therapeutics, CTX112, demonstrates that these multiplexed edits can produce cells with expansion and potency comparable to autologous therapies. In trials for relapsed or refractory B-cell malignancies, CTX112 induced complete responses in patients who had previously failed other advanced therapies. Solid tumors, examples lung, breast, glioblastoma, represent 90% of cancer cases, but have largely resisted CAR T therapy due to physical barriers and a hostile, immunosuppressive tumor microenvironment, TME. CRISPR is being used to armor T cells against these defenses. Breaking the barriers. PD-L1 IL-12 fusion secreting cells. Researchers at USC Norris Comprehensive Cancer Center engineered CAR T cells to secrete a fusion protein of IL-12, a potent immune activator, and a PD-L1 blocker. This design localizes the immune boost to the tumor site, avoiding the systemic toxicity usually associated with IL-12. In mouse models of prostate and ovarian cancer, this strategy successfully shrunk tumors that were resistant to standard CAR T therapy. Targeting PD-L1 directly. A Mayo Clinic study utilized CAR T cells targeting PD-L1 itself, a protein expressed on both tumor cells and the surrounding stromal cells. This dual targeting approach allows the T cells to destroy the tumor's support structure. Notably, these cells demonstrated the ability to cross the blood-brain barrier to treat glioblastoma in preclinical models. New targets. Trials initiating in 2025 will target antigens like Glypican-3, GPC3, and CD70, CTX131, in solid tumors such as renal cell carcinoma and liver cancer. These cells are heavily engineered with CRISPR to resist the metabolic starvation and inhibitory signals typical of the solid tumor interior. Cystic fibrosis, the challenge of delivery. While blood diseases can be treated by removing cells, editing them, and putting them back, cystic fibrosis, CF, requires editing cells deep within the lungs, a formidable delivery challenge. CF is caused by mutations in the CFTR gene, leading to thick, sticky mucus that clogs airways and traps bacteria. The lung is evolved to keep foreign objects out. A thick layer of mucus and the tight junctions of the airway epithelium make it incredibly difficult for gene editing tools like CRISPR machinery packaged in lipid nanoparticles, or LNPs, to reach the basal stem cells necessary for a durable cure. If only the surface cells are edited, they will naturally slough off and die within months, requiring repeated treatments. Viral vectors. ADAVs are efficient delivery vehicles but have a limited cargo size, approximate 4.7 kb. The CFTR gene is large, 4.5 kb, and adding the Cas9 gene and gRNA exceeds the capacity of a single ADAV. Researchers are exploring split-vector approaches, where the editing machinery is divided between two viruses that must co-infect the same cell. Lipid nanoparticles, LNPs. LNPs are safer than viruses, but traditionally accumulate in the liver. Novel LNP formulations decorated with lung-targeting ligands are in development. Snippets indicate intense research into optimizing these vehicles to penetrate the mucus layer. Stem cell editing. Another strategy involves harvesting airway basal stem cells, editing them ex vivo, similar to the SCD model, and retransplanting them. Stanford researchers achieved a 70% restoration of CFTR function in cultured human cells using this method, though engraftment in the lung remains a hurdle. The N of 1 revolution. Personalized genetic medicine. CRISPR's programmability allows for the rapid development of bespoke medicines for ultra-rare diseases, conditions affecting so few people that pharmaceutical companies typically ignore them. The most compelling example of this new paradigm occurred in 2024-2025 with baby KJ, an infant born with carbamyl phosphate synthetase 1 deficiency, a fatal metabolic disorder. The timeline. In an unprecedented feat of regulatory and scientific agility, a custom base editing therapy was designed, manufactured, and FDA cleared within 7 months of the child's birth. The therapy. A base editor carried by an LNP was infused systemically to target the liver cells, hepatocytes. The outcome. After 3 doses, KJ showed stabilized ammonia levels and reduced reliance on restrictive diets and medications. This N of 1 trial proves that the regulatory pathway can be adapted for hyper-personalized medicines. Tay-Sachs disease. For late-onset Tay-Sachs disease caused by HEXA gene mutations, researchers are using base editing to restore enzyme activity. Preclinical data suggests that restoring even 10% of enzyme function can halt neurodegeneration. Huntington's disease. This dominant genetic disorder requires silencing the toxic mutant HTT allele. The blood-brain barrier, BBB, is the primary obstacle. Innovative strategies using focused ultrasound to temporarily disrupt the BBB, allowing CRISPR-loaded nanoparticles to enter the brain, have shown success in animal models. This combination of physical and genetic engineering represents a cutting-edge frontier in neurology. Human augmentation and longevity, the speculative frontier. As CRISPR masters the art of fixing deficits, the conversation inevitably shifts to enhancement, pushing human biology beyond its baseline. The line between therapy and enhancement blurs with vaccine-like gene edits. The infamous 2018 case of He Jiankui, who edited the CCR5 gene in embryos to confer HIV resistance, remains a cautionary tale. In 2024, He Jiankui returned to research, now focusing on Alzheimer's and Duchenne muscular dystrophy in embryos, stating he is proud of his past work. While the global scientific community condemns germline editing, heritable changes, somatic editing for disease immunity is advancing. Therapies knocking out PCSK9 to permanently lower cholesterol or CCR5 for HIV cure are effectively forms of biological enhancement, conferring supernormal resistance to environmental threats. Aging is increasingly viewed as a malleable biological process driven by epigenetic drift and cellular senescence. Epigenetic reprogramming. Researchers are using dead Cas9 fused to epigenetic activators, CRISPR AI, to transiently express Yamanaka factors, OSKM. This process can reset the epigenetic clock of a cell, restoring youthful gene expression patterns without stripping the cell of its identity. Recent studies have demonstrated rejuvenation effects in primate cells and mice using this technique. Senolysis. CRISPR is also being designed to hunt and kill senescent zombie cells that accumulate with age and drive systemic inflammation. By targeting promoters active only in senescent cells, like P16, Gene circuits can trigger apoptosis specifically in these aged cells, improving health span. Agricultural revolution, food security in a changing climate. CRISPR's impact extends well beyond the clinic. In agriculture, it offers a tool to adapt crops to climate change and reduce reliance on chemical inputs. Unlike transgenic GMOs, which introduce foreign DNA, CRISPR often involves simple deletions or edits that could theoretically occur in nature, leading to a friendlier regulatory environment. Saving chocolate. Cacao plants are devastated by Phytophthora, black pod disease. Penn State researchers utilized CRISPR to delete the TCNPR3 gene, which acts as a suppressor of the plant's immune system. The edited plants demonstrated significantly enhanced resistance to the pathogen without the introduction of any foreign DNA, preserving the specific flavor profiles of the cacao varieties. Gluten-free wheat. Attempts to edit the gliadin genes in wheat to remove the epitopes that trigger celiac disease are advancing. This involves multiplexed editing to knock out roughly 30 to 40 gene copies simultaneously, a feat uniquely suited to CRISPR. Gamba tomatoes. In Japan and now entering other markets, CRISPR-edited tomatoes with elevated levels of gamma-aminobutyric acid are sold as functional foods to help lower blood pressure. Pitless fruit. Companies like Pairwise are marketing CRISPR-edited greens to remove bitterness and are developing seedless berries to increase consumer appeal. A major hurdle for agricultural biotechnology has been the European Union's strict GMO regulations. However, in 2025, the landscape shifted significantly. The EU Council agreed on a mandate for new genomic techniques, NGTs. This legislation creates a two-tiered system. Category 1 NGTs. Plants with edits that could have been achieved via conventional breeding. These will be treated like conventional crops, no special labeling. Category 2 NGTs, plants with more complex, transgenic-like edits. These will remain under stricter regulation. This regulatory evolution is expected to unleash a wave of innovation in European agriculture, aligning it more closely with the US and Asian markets. The economics of gene therapy. Innovation versus access. The scientific triumph of CRISPR is colliding with the economic reality of healthcare systems. Casgevy launched with a list price of $2.2 million per patient. For rare diseases, such prices are rationalized by the lifetime value of the cure, offsetting decades of hospitalizations and chronic care. However, for conditions like sickle cell disease, which predominantly affects populations in low-income countries like sub-Saharan Africa or marginalized communities in the US, this price tag is a formidable barrier. The upfront cost creates a liquidity crisis for payers, particularly state Medicaid programs. To democratize access, the cost of delivery must come down. The current ex vivo model, hospitalization, chemotherapy, stem cell transplant, is inherently expensive and unscalable globally. The Bill and Melinda Gates Foundation and the NIH have committed $200 million to develop in vivo gene therapies for HIV and SCD. The goal is a single-shot cure, a viral vector or LNP that can be administered in an outpatient setting, locating and editing the target cells inside the body. This would eliminate the need for specialized transplant centers and bring the cost down to a level feasible for global health distribution. Navigating the genome. We have entered the age of the engineered genome. CRISPR has delivered on its initial promise, providing functional cures for devastating genetic diseases and reshaping the landscape of agricultural biotechnology. The shift from blunt DNA cutting to precise base and prime editing is resolving early safety concerns, while the transition from ex vivo to in vivo delivery promises to solve the accessibility crisis. But the technology is moving faster than our ethical and economic frameworks can adapt. As we stand on the threshold of human enhancement and germline intervention, the choices made in the next decade by regulators, scientists, and society will determine whether CRISPR becomes a tool for widening inequality or a universal instrument of human liberation. The horizon is bright with hope, but it is a horizon we must approach with vigilance, ensuring that the power to rewrite life is exercised with wisdom equal to its potency. CRISPR, the molecular scissors explained. An exhaustive analysis of the programmable genome. The dawn of the genetic searchlight. In the grand tapestry of biological evolution, few mechanisms illustrate the relentless arms race between predator and prey as vividly as the immune systems of prokaryotes. For billions of years, bacteria and archaea have waged a silent molecular war against their viral nemesis, the bacteriophages. From this primordial conflict emerged a defense system of such elegant simplicity and ruthless efficiency that its discovery has fundamentally altered the trajectory of human science. This system is known as CRISPR. Clustered, regularly interspaced, short palindromic repeats. Often conceptualized through the analogy of molecular scissors, the CRISPR-Cas9 system represents the ultimate convergence of biological function and engineering utility. If one imagines the vast, distinct landscape of the genome as a dark, uncharted territory, the CRISPR system functions as a highly sophisticated global positioning system, GPS, capable of navigating billions of base pairs to locate a single, specific coordinate. Once arrived, the Cas9 enzyme acts as the precise cutting tool, the molecular scissors, severing the strands of the double helix to initiate a modification. But the true genius of the system lies in its guidance component, the guide RNA, gRNA. This molecule acts as a searchlight, illuminating the target sequence with thermodynamic precision, allowing the Cas9 machinery to distinguish the signal of the target from the noise of the surrounding genome. To fully appreciate the magnitude of CRISPR technology, one must move beyond surface-level analogies and descend into the atomic and mechanistic realities of the system. This part provides an exhaustive deconstruction of CRISPR-Cas9. It traces the intellectual lineage from the salt marshes, where the repeats were first noticed, to the high-tech laboratories where they are now used to rewrite the code of life. It explores the intricate structural ballet of the Cas9 enzyme, the physicochemical forces that drive DNA melting and R-loop formation, and the revolutionary search and replace technologies that are evolving beyond the simple cut. By synthesizing data from structural biology, genetics, and biophysics, we reveal how a primitive bacterial memory bank became the most powerful tool in the modern geneticist's arsenal. The historical Odyssey, from anomalous repeats to adaptive immunity. The history of CRISPR is a testament to the importance of curiosity-driven research. It is a narrative punctuated by serendipitous discoveries, where researchers investigating unrelated phenomena, from saline tolerance in archaea to the fermentation of yogurt, slowly piece together a puzzle that would eventually reveal a programmable immune system. The story begins in 1987 at Osaka University, where Yoshizumi Ishino and his team were sequencing the IAP gene in Escherichia coli. In the 3' flanking region of this gene, they observed something bizarre: a set of 5 homologous sequences of 29 nucleotides, arranged as direct repeats, separated by 32 nucleotide spacer sequences of unknown origin. At the time, the biological significance of these clustered repeats were entirely opaque. They were an anomaly, a genetic stutter in the bacterial code. Throughout the 1990s, as sequencing technology improved, similar repeat clusters were identified in a diverse array of bacteria and archaea. In 1993, Francisco Mojica, working at the University of Alicante in Spain, characterized these loci in the halophilic archaeon Halopharax mediterranei. Mojica was struck by the ubiquity of the structures and their conservation across distant species. It was Mojica who, in correspondence with Ruud Janssen, coined the term CRISPR in 2002. The breakthrough came in 2005. Mojica, alongside independent groups led by Alexander Bolotin and others, performed bioinformatic analyses on the unique spacer sequences sandwiched between the repeats. They made a startling discovery. The spacers were not random. They were, in fact, perfect matches to sequences found in bacteriophages, viruses that infect bacteria, and plasmids. This observation led to the revolutionary hypothesis that CRISPR was an adaptive immune system. The bacterium was capturing snippets of viral DNA and storing them in its own genome as a genetic memory of past infections. The hypothesis of adaptive immunity moved from bioinformatics to experimental reality in 2007, driven by the dairy industry. Scientists at Danisco, led by Philippe Horvath and Rodolphe Berengow, were studying Streptococcus thermophilus, a bacterium essential for yogurt and cheese production but highly susceptible to phage attacks. In a landmark paper published in Science, they demonstrated that Streptococcus thermophilus strains that survived phage infection had acquired new spacers in their CRISPR loci derived from the attacking phage genome. Furthermore, they showed that deleting these spacers abolished resistance, while inserting new ones conferred resistance. This was the first direct evidence that CRISPR was a memory bank for the immune response. With the function established, the race was on to understand the mechanism. How did these stored sequences actually stop a virus? The RNA Guide, 2008. John van der Oost and colleagues demonstrated that the CRISPR array is transcribed into long RNA molecules, pre-cRNA, which are then processed into small CRISPR RNAs, cRNAs. These cRNAs guide a complex of Cas proteins to the target viral DNA. This confirmed the Searchlight role of the RNA. The target is DNA, 2008. Luciano Marafini and Eric Sontheimer showed that the CRISPR system targets DNA, not RNA, unlike eukaryotic RNA interference. This was a crucial distinction that paved the way for genome editing applications. The Dual RNA System, 2011. Emmanuel Charpentier, studying Streptococcus pyogenes, discovered a second essential RNA component, the trans-activating CRISPR RNA, tacRNA. She found that tracker RNA pairs with the repeat sequences of the pre-cRNA and is essential for its processing and for the function of the Cas9 enzyme. The pivotal moment for biotechnology arrived in 2012. Jennifer Doudna and Emmanuel Charpentier published a seminal paper in Science demonstrating that the Cas9 enzyme could be programmed with a synthetic RNA to cut any DNA sequence in vitro. Crucially, they fused the cRNA and tracker RNA into a single chimeric molecule, the single guide RNA, sgRNA. This simplified the three-component system, Cas9, cRNA, tracker RNA, into a two-component system, Cas9, sgRNA, creating the plug-and-play tool we use today. Shortly thereafter, in 2013, Fang Zhang and George Church independently demonstrated the application of this system in mammalian cells. Zhang's work was particularly notable for identifying the need for codon optimization and nuclear localization signals, NLSs, to ensure the bacterial protein could function within the complex environment of the eukaryotic nucleus. The bacterial arsenal, adaptation, and interference. To utilize CRISPR as a tool, we must first understand it as a biological system. In its natural context, CRISPR immunity occurs in three distinct stages: adaptation, acquiring the memory; expression, creating the searchlight; and interference, cutting the target. While gene editing focuses largely on interference, the adaptation stage contains the logic for the system's specificity. Adaptation is the process of acquiring new spacers from invading viruses. This step is mediated by the highly conserved Cas1-Cas2 integrase complex. The Cas1-Cas2 complex. Structurally, two dimers of Cas1 flank a central Cas2 dimer, forming a butterfly-like architecture. This complex acts as a molecular ruler, binding to the viral DNA and ensuring that the captured fragment is of a precise length, typically 30 BP in type 2 systems. This precise sizing is critical. If the spacer is too long or too short, it will not fit into the CRISPR array correctly. The role of CSN2. In type 2 systems, like SP-Cas9, Cas1 and Cas2 are assisted by an accessory protein called CSN2. Cryo-electron microscopy, cryo-EM, studies have revealed that CSN2 forms a homotetrameric ring structure. This ring acts similarly to a sliding clamp in DNA replication, encircling the DNA and bridging the Cas1-Cas2 complex. This supercomplex, Cas9, Cas1, Cas2, CSN2, stabilizes the pre-spacer DNA and facilitates its integration into the leader proximal end of the CRISPR array. PAM selection in acquisition. A critical aspect of adaptation is that the machinery must select a pre-spacer that is flanked by a protospacer adjacent motif, or PAM. If the system captured a random sequence without a PAM, the resulting guide RNA would be useless because Cas9 requires the PAM to cut. Therefore, Cas9 itself is involved in the acquisition complex to verify the presence of a PAM on the viral DNA before it is captured. After the spacer is integrated, the bacterium must manufacture the guides. Transcription. The entire CRISPR array is transcribed into a long precursor RNA, pre-CRISPR RNA. Hybridization. The transactivating RNA, tracer RNA, is transcribed separately. It contains a sequence perfectly complementary to the repeat sequences of the pre-CRISPR RNA. The tracer RNA finds the repeats on the pre-CRISPR RNA and binds to them, forming double-stranded RNA segments. Processing. A host enzyme, RNase 3, recognizes these double-stranded RNA regions and cleaves them. This releases individual mature CRISPR RNAs, each consisting of a single spacer, the viral memory, and a partial repeat still annealed to the tracer RNA. This dual RNA structure is what binds to Cas9. The Cas9 RNA complex now patrols the cell. It faces a dangerous paradox. The bacterium's own genome contains the spacer sequences in the CRISPR array that match the guide RNA. Why doesn't Cas9 commit suicide by cutting its own genome? The solution lies in the PAM. Target, viral DNA. The viral target sequence, protospacer, is flanked by the PAM, e.g., NGG. Cas9 recognizes the PAM and cuts. Self, CRISPR DNA. The spacer sequence in the CRISPR array is flanked by the repeat sequence, which does not contain the PAM. Because Cas9 has an absolute requirement for the PAM to initiate binding and cleavage, it effectively discriminates between the non-self viral target, PAM present, and the self-storage locus, PAM absent. This simple digital logic, PAM versus no PAM, is the safeguard of the bacterial genome. The atomic anatomy, structure of the Cas9 machine. The Cas9 enzyme is not a static molecular rock. It is a dynamic, multi-domain machine that undergoes massive conformational rearrangements. High-resolution crystal structures of Streptococcus pyogenes Cas9, or SP-Cas9, have revealed a bilobed architecture that is central to its function as a programmable nuclease. Cas9 is divided into two primary lobes, connected by a linker region. The recognition, or REC lobe. This lobe is the GPS interface. It is primarily responsible for binding the guide RNA and scanning the DNA target. It is subdivided into three domains: REC1, REC2, and REC3. REC1 is the largest domain and serves as the primary anchor for the repeat-anti-repeat duplex of the sgRNA. It holds the handle of the Searchlight. REC2 and REC3 are flexible and play a crucial role in proofreading. They interact with the RNA-DNA heteroduplex, the hybrid formed when the guide binds the target, to verify complementarity. If the match is not perfect, these domains prevent the enzyme from transitioning to the active state. The nuclease lobe. This lobe is the scissors. It contains the catalytic machinery required to sever the DNA backbone. It houses two distinct nuclease domains, HNH and RUVC, and the PAM interacting domain. Connecting the REC and NUC lobes is an arginine-rich structure called the bridge helix. This helix acts as a hinge, regulating the relative movement of the two lobes. Upon binding the guide RNA, the bridge helix undergoes a conformational shift that creates a central channel, a positively charged groove where the DNA target will eventually sit. This prepares the scissors for the arrival of the substrate. Unlike traditional restriction enzymes that often use a single active site to cut both strands via dimerization, Cas9 functions as a monomer with two distinct active sites. This dual nuclease architecture allows for precise control over the cleavage of each strand. The HNH domain, named for its characteristic histidine-aspargine-histidine motif, this domain structurally resembles homing endonucleases like T4 endonuclease VII. It is responsible for cleaving the target strand, which is the DNA strand that is complementary to the guide RNA. Structurally, the HNH domain is highly flexible. It must swing into position to access the DNA phosphate backbone only after the target has been verified. The RUVC domain, named after the E. coli RUVC holiday junction resolvase, this domain cleaves the non-target strand, which is the DNA strand that is displaced by the RNA. It uses a two-metal ion mechanism, typically involving magnesium, to catalyze the hydrolysis of the phosphodiester bond. The RUVC domain is structurally discontinuous, formed by three separate motifs, RUVC1, 2, and 3, that fold together to form the active site. The engineering of the sgRNA by Doudna, Charpentier, and Jinek involved fusing the 3' end of the cRNA to the 5' end of the tracker RNA via a synthetic tetraloop. The spacer, or the Searchlight, the first 20 nucleotides at the 5' end. This is the user-programmable sequence that defines the target. The scaffold, the remaining sequence, the tracker RNA and cRNA hybrid, folds into a series of hairpins, stem loops 1, 2, and 3 that bind tightly to the REC and NUC lobes, stabilizing the complex. Without this scaffold, Cas9 remains an inert protein, unable to bind DNA. The physics of the search. Mechanism of action. How does a single molecule find a unique 20 base pair sequence in a human genome of 3.2 billion base pairs? The process involves a combination of 3D diffusion, thermodynamic interrogation, and kinetic proofreading. It is a searchlight that does not just scan, it physically pries open the genome to check for a match. The Cas9 sgRNA complex, or RNP, diffuses randomly through the nucleus. It does not unzip every piece of DNA it touches. The energy cost would be prohibitive. Instead, it uses the PAM as a first-pass filter. The PI domain. The PAM interacting domain at the C-terminus of Cas9 contains a specific pocket that recognizes the 5' NGG 3' motif. Two arginine residues, arginine 1333 and arginine 1335, act as fingers that probe the major groove of the DNA. Hydrogen bonding. These arginines form hydrogen bonds with the guanine bases of the PAM on the non-target strand. This interaction is rapid and reversible. If Cas9 collides with DNA lacking a PAM, it dissociates in milliseconds. If it finds a PAM, it pauses, tethering itself to the DNA. This mechanism reduces the search space by orders of magnitude, as Cas9 effectively ignores any sequence not flanked by an NGG. Once the PAM is bound, the DNA double helix must be separated, melted, to allow the guide RNA to base pair with the target strand. For years, the consensus was that the Cas9 protein itself physically bent the DNA, forcing the strands apart. However, recent research has nuanced this view. The phosphate lock. Residues in the Cas9 protein, lysine 1107 and serine 1109, interact with the phosphate backbone of the DNA strand immediately upstream of the PAM. This interaction, known as the phosphate lock, creates a kink in the DNA, mechanically destabilizing the helix. RNA-mediated intercalation. New molecular dynamic simulations and biophysical data suggest that the guide RNA plays a more active role than previously thought. The first nucleotide of the guide RNA in the seed region appears to intercalate into the DNA duplex, physically inserting itself between the DNA bases. This RNA intrusion disrupts the stacking interactions of the DNA base pairs, lowering the energy barrier for strand separation. This suggests the searchlight is not passive illumination, but an active physical probe. Consensus. The melting is likely a cooperative effort. The protein, PAM binding plus phosphate lock, destabilizes the helix, and the RNA, intercalation, capitalizes on this instability to invade the duplex. As the DNA melts, the guide RNA begins to base pair with the target strand, displacing the non-target strand. This structure, an RNA-DNA hybrid plus a single strand of displaced DNA, is called an R-loop. Directional zipping. R-loop formation is strictly directional. It initiates at the seed sequence, the 8 to 10 nucleotides adjacent to the PAM, and propagates towards the 5' end of the target, distal to the PAM. The seed checkpoint. The seed region is the high-fidelity zone. Thermodynamics dictate that if the RNA does not match the DNA perfectly in this region, the R-loop collapses immediately and Cas9 falls off. This is why mismatches in the seed region are rarely tolerated. Breathing motions. The Cas9 cleft undergoes micro-expansions and contractions that facilitate the base-by-base zipping of the RNA and DNA. This dynamic movement helps the system overcome energy barriers during the formation of the full 20-base pair hybrid. Binding the target is not enough. Cas9 must be activated to cut. The enzyme possesses a built-in safety mechanism known as the conformational checkpoint. The HNH swing. In the inactive state, the HNH nuclease domain is positioned away from the DNA. As the R loop reaches its full length, extending past the seed, the REC3 domain senses the formation of the distal RNA-DNA hybrid. Allosteric signal. This sensing triggers a massive conformational change. The Rebe lobe rotates by approximately 15 degrees relative to the nuke lobe. This rotation releases the HNH domain from its locked state, allowing it to swing 25 angstroms into the catalytic center, docking precisely onto the target strand cleavage site. Coupled activation. The movement of the HNH domain transmits a signal to the RUV-C domain, activating it as well. This ensures concerted cleavage. Both strands are cut simultaneously, or nearly so, to generate a clean double-strand break, DSB. Implication: if the guide RNA has mismatches at the distal end, far from PAM, the R loop may form, but the REC3 domain may not trigger the conformational change. The Cas9 will bind, but not cut. This binding without cutting is a significant source of off-target behavior in some contexts, where Cas9 acts as a blockade rather than a nuclease. From bacteria to bench, the engineering of a tool. The transition of Cas9 from a bacterial immune effector to a mammalian genome editing tool required overcoming significant biological barriers. Bacteria and humans are separated by billions of years of evolution, differing in cellular compartmentalization, genetic coding, and DNA accessibility. The most obvious difference is that human DNA is sequestered in the nucleus, while bacterial DNA floats in the cytoplasm. Cas9 is a large protein, about 160 kilokadaltons, and cannot passively diffuse through the nuclear pore complex. To solve this, researchers appended nuclear localization signals, or NLS, to the Cas9 protein. These short peptide sequences, often derived from the SV40 virus, can act as shipping labels. They are recognized by important proteins in the eukaryotic cytoplasm, which actively transport the Cas9 cargo into the nucleus. While the genetic code is universal, AUG codes for methionine in all life, different organisms prefer different synonymous codons for the same amino acid. Streptococcus pyogenes is an AT-rich organism and prefers codons ending in A or T. Human cells are more GC-rich. If the native bacterial Cas9 gene is introduced into a human cell, the human ribosome may stall or terminate translation due to the scarcity of the appropriate tRNAs for the bacterial codons. To ensure robust expression, the Cas9 gene was codon optimized. The DNA sequence was rewritten to use the codons most frequently used by human cells without changing the amino acid sequence of the protein. Getting the Cas9 and sgRNA into the cell is a major engineering challenge. Several modalities exist. Plasmids, circular DNA encoding Cas9 and sgRNA. The cell must transcribe and translate them. This provides sustained expression but increases the risk of off-target edits due to the prolonged presence of the nuclease. mRNA, delivering Cas9 mRNA and sgRNA. The cell translates the protein immediately. This is transient. The mRNA degrades quickly, reducing off-target risks. Ribonucleoprotein, or RNP. Purified Cas9 protein pre-complexed with synthetic sgRNA. This is the hit-and-run method. The complex is active immediately upon entry and is degraded within hours. This yields the highest specificity and is the preferred method for clinical applications. Viral vectors, adeno-associated viruses, are used to deliver the genetic instructions to specific tissues in the body. However, SP-Cas9 is large and barely fits into an AAV. This has driven the search for smaller Cas9 orthologs, such as SA-Cas9, Staphylococcus aureus Cas9, which is significantly smaller, about 1,053 amino acids versus 1,368 for SP-Cas9, and fits easily into viral vectors. The cellular response. Repair pathways. Cas9 is, strictly speaking, a damaging agent. It breaks the DNA. The editing is the result of the cell's desperate attempt to survive this damage. The outcome of a CRISPR experiment is dictated not by the scissors, but by the cellular repair crew that fixes the cut. NHEJ is the dominant repair mechanism in mammalian cells, active throughout the cell cycle, G1, S, G2, M. It is a smash-and-grab repair. The cell assumes the two broken ends belong together and stitches them back up, often without a template. The mechanism. The Cu-SO70, Cu-80 heterodimer rapidly binds to the broken DNA ends, protecting them from degradation. Cu recruits the catalytic subunit DNA-PKCS, which in turn recruits Artemis, a nuclease, and the XRCC4 DNA ligase 4 complex. The indel. During this process, ends may be slightly chewed back, or small bases may be added by polymerases. This results in indels, insertions, and deletions at the junction. The knockout. If the indel creates a frame shift, for example, adding one or two bases in a protein-coding gene, the downstream sequence becomes nonsense and a premature stop codon is usually generated. This destroys the gene's function, a knockout. HDR is the photocopier pathway. It uses a homologous sequence, usually the sister chromatid, as a template to copy the missing information across the break. This pathway is generally error-free, but is restricted to the S and G2 phases of the cell cycle, when the sister chromatid is available. The opportunity. Researchers hijack this pathway by flooding the cell with a donor template, plasmid or single-stranded oligonucleotide. This donor contains the desired edit, like correcting a disease mutation, flanked by homology arms that match the sequence around the cut. The mechanism. The MRN complex, Menahan Rad50 NBS, detects the break. C-tip promotes the resection of the 5' ends, leaving 3' single-stranded overhangs. These overhangs are coded by RPA, which is then replaced by Rad51. The Rad51 filament invades the homologous template, the donor, forming a D-loop and copies the sequence. The challenge. In most cells, NHEJ outcompetes HDR. The efficiency of precise editing, knock-in, is often low, 1 to 10%. Researchers try to boost HDR by inhibiting NHEJ, for example, using small molecules to block DNA ligase 4, or by synchronizing cells in the S phase. Also called alternative NHEJ, this pathway relies on short microhomologies, 5 to 25 BP, exposed by end resection. Mechanism. PARP1 and DNA ligase 3, instead of ligase 4, drive this pathway. The single-stranded overhangs anneal at the microhomology regions. the non-homologous flaps are clipped off. Result: this always results in a deletion of the sequence between the microhomologies. While error-prone, it is predictable. If researchers design sgRNAs to target sites with flanking microhomologies, they can induce specific, reproducible deletions, which is useful for studying regulatory elements. The next generation, beyond the scissors. While the scissors model revolutionized biology, creating double-strand breaks, or DSBs, is hazardous. DSBs can lead to p53 activation, cell death, large deletions, or chromosomal translocations if two chromosomes are cut simultaneously. To address this, the field has evolved towards nicking and writing technologies that avoid DSBs entirely. By mutating a single residue in one of the nuclease domains, such as D10A in RUVC or H840A in HNH, Cas9 is converted into a nickase, or Cas9N. It cuts only one strand. Double nicking. Researchers use two paired nickases, targeting opposite strands separated by a short offset. This creates a staggered cut. Both strands are broken, but the overhangs stimulate repair. Crucially, if one nickase binds off target, it creates a single-strand nick, which the cell repairs faithfully using base excision repair, or BER, without mutation. This effectively eliminates off-target mutations. Developed by David Liu's lab, base editors, or BEs, allowed the direct chemical conversion of one DNA letter to another without cutting the backbone. Cytosine base editors, or CBEs, a dead or dCas9 or nickase is fused to a cytidine deaminase enzyme, such as ApoBec. Mechanism: the Cas9 opens the DNA R loop. The deaminase accesses the single-stranded non-target strand and chemically deaminates cytosine, or C, into uracil, or U. U has the base pairing properties of thymine, or T. Upon DNA replication, the cell pairs the U with an adenine, or A, effectively converting a CG pair to a TA pair. Adenine base editors. No natural enzyme deaminates adenine in DNA. Liu's lab evolved a tRNA deaminase, or TADA, to accept DNA substrates. Mechanism: the ABE converts adenine, or A, to inosine, or I. Inosine is read by the polymerase as guanine, or G. This converts an AT pair to a GC pair. Significance: these tools can correct 60% of known human pathogenic point mutations, or SNPs, with high efficiency and no DSBs. Base editors are limited to transitions, C to T, A to G, and cannot correct transversions, such as A to T, or insert or delete bases. Prime editing, also from the Liu Lab, 2019, solves this. The complex: a Cas9 nickase is fused to a reverse transcriptase. The PEG RNA. The guide RNA is extended at the 3' end to form a prime-editing guide RNA, or PEG RNA. This extension contains two critical sections: primer binding site, or PBS, complementary to the nicked DNA strand; reverse transcriptase template, or RTT, contains the desired edit sequence. The mechanism: the Cas9 nicks the DNA. The PBS hybridizes to the nicked DNA flap. The reverse transcriptase grabs the nicked 3' end and uses the RTT as a template to polymerize or write the new DNA sequence directly onto the end of the genomic strand. The cell eventually incorporates this new flap and excises the old sequence. Analogy: if Cas9 is scissors, and base editors are a red pen. Prime editing is a word processor. It can search for a sequence and replace it with any other sequence. Insertions, deletions, all 12 base-to-base conversions. Implications, applications, and future horizons. The Searchlight of CRISPR has illuminated paths in medicine and agriculture that were previously invisible. Hemoglobinopathies. The first approved CRISPR therapy, Casgevy, treats sickle cell disease and beta thalassemia. It uses CRISPR to disrupt the enhancer of the BCL11A gene in patient stem cells. BCL11A is the brake on fetal hemoglobin. Cutting the brake reactivates fetal hemoglobin, compensating for the defective adult blood cells. In vivo editing. For diseases like transthyretin amyloidosis, ATTR, CRISPR lipid nanoparticles are injected directly into the bloodstream. They home to the liver, where Cas9 knocks out the misfolding TTR gene. This represents a one-and-done, lifelong cure. The discovery of other Cas enzymes like Cas13, which targets RNA, and Cas12, which targets DNA, led to a new class of diagnostics. These enzymes have a property called collateral cleavage. Upon finding their specific target, e.g., a COVID-19 viral sequence, they go into a frenzy, shredding any nearby nucleic acids. Mechanism. By adding a reporter, a fluorescent tag linked to a quencher, to the reaction, the collateral cleavage cuts the link, releasing the fluorescence. This creates a rapid paper strip test that lights up in the presence of the virus. CRISPR allows for the creation of crops that are resistant to drought, pests, or browning without introducing foreign DNA, or transgenics. Because the final plant contains only the edited sequence and not the Cas9 machinery, these crops are often regulated differently than traditional GMOs. Examples include non-browning mushrooms and mildew-resistant wheat. The power to rewrite the code of life carries immense responsibility. Germline editing. Editing somatic cells, body cells, affects only the patient. Editing germline cells, sperm, eggs, or embryos, changes the genetics of all future generations. The CRISPR babies scandal of 2018 highlighted the risks of premature application, including off-target mutations and mosaicism, where only some cells are edited. Off-target effects. While engineered variants like SP-Cas9HF1 have reduced off-target activity to near background levels, the absolute validation of zero off-targets remains a challenge for regulatory approval. To define CRISPR-Cas9 merely as molecular scissors is to capture only its initial crude capability. It is, in reality, a programmable DNA binding platform. It is a homing device, a universal GPS that can drag any molecular cargo to any genomic address. Whether that cargo is a nuclease to cut, Cas9, a deaminase to mutate, base editor, a polymerase to write, prime editor, a transcription factor to activate, CRISPR-AI, or a fluorescent protein to tag, CRISPR imaging, the core principle remains the same: the interaction between the guide RNA Searchlight and the DNA target. By separating the hardware, the Cas9 protein, from the software, the guide RNA, evolution created a modular system that was uniquely primed for human repurposing. We no longer need to build a new machine for every job. We simply update the software. As we move from the era of cutting to the era of writing, the searchlight of the guide RNA continues to illuminate new possibilities, promising a future where the genetic causes of disease are not just treated, but corrected at their source. The scissors were just the beginning. The programmable genome is the destination.